We're going to look at two more separation techniques. This is part of section 5.5. And last time we looked at a process called distillation, which separated two liquids. This is going to be the process of separating two dissolved solids. And this process is through a process called chromatography. And particularly we're going to be looking at what's called paper chromatography. Paper chromatography. So imagine that you have two solids. They are now dissolved inside some form of liquid. And paper chromatography comes in many different shapes. The most common type of paper chromatography is on what's called a chromatography strip. And this strip is a couple of centimeters wide, and it tapers down to a point right here. And this is often put inside of a test tube or some other container. And what you do is you take your two solids that you're trying to separate and you put them and lay them down right here in a band toward the bottom of your chromatography strip. So right here, this is your paper chromatography. This is actually, it's made out of like a filter paper. And this is inside of a test tube or some other container. And this container is then going to have some type of liquid in it. And this liquid at the bottom is called the solvent. And these materials that dissolve in the solvent will be able to be carried by this liquid. So you have a test tube or container that has this liquid, and you have this chromatography strip that has your dissolved solids that are ready to be separated. You put this filter paper in this container, and what happens is the filter paper begins to suck up this liquid like a sponge. And the liquid begins to move up towards the dissolved solids. And what happens is then it reaches these dissolved solids, and over time it's going to start to push these solids up the paper. So this is a kind of a before snapshot of what's going to be happening. Let's show what's going to be happening in the aftermath. So you have your container. Here's your chromatography strip. And the liquid's going to be a little bit lower because a lot has run through the paper chromatography, and it's pushed those solids up, maybe to here and here. So it's pushed this solid to here, and it's pushed this solid up to here. And then you have successfully separated those two materials. So the question is, why did they separate at two different distances? What is or are the factors that cause this to separate differently? So the distance that this dissolved solid moves, the distance of movement, it depends on many factors. The distance of movement depends on three factors. The first factor is the size of the dissolved solid. The size of the dissolved solid. What's it going to be easier to move through a material? A small substance or a large substance? Now imagine you have a sponge with all these little pores and holes. What's going to make it through those tunnels a lot easier? And it's going to be a smaller substance. So smaller substances will be faster. Smaller are faster. The larger particles will move more slowly because they're going to get stuck. Another factor depends upon its chemical composition, and it's how it sticks to the paper. How much it sticks to the paper. This is a concept called adsorption, not absorption, but adsorption. Note the D. Adsorption is how much that it sticks to the paper. So the first is based upon size. Smaller will be able to easily get through the tunnels of the paper. But this is chemically, how much is it going to stick to the paper? And the third one is the solvent that's being used, this liquid. It's going to be how much it's pushed by the liquid. How much it's pushed by the liquid solvent. So we'll have a lab on that to see this process of separating two dissolved solids. This is called paper chromatography because it was often used in plant pigments. Green plants don't just have one pigment, they actually have multiple pigments. And you can crush the pigments of a leaf into this paper, this filter paper, and you have your liquid solvent. And what will happen is the liquid solvent will push the pigments up the paper, and you can actually separate your pigments based upon their various colors. And you'll see various types of greens and yellows and oranges in land plants. And that's why it's called chromatography, because chroma means color and graph means to chart. It's a little chart of the colors. It's a separation of the colors. So we'll see that also in our lab. So the three main factors that will affect how much it's going to move, again, are the size of the particle, smaller will go faster, how much it sticks to the paper, which is called the adsorption, and how much it's being pushed by the solvent liquid. The third factor that we're going to take a look at, or a third separation technique, is going to be taking a look at solids and liquids. We first looked at distillation, which separated two liquids. Then we took a look at paper chromatography to separate two dissolved solids. This next one is going to be separating solids and liquids. Solids and liquids. And we can do this with a process called filtration. We're simply going to filter our material. So imagine that you have a mixture of sand and water. What we can do is we can get a funnel. So here's our funnel. And we're going to be putting a filter to cover the funnel, much like the filter paper of this. So we'll have a filter, and we're then going to have a container underneath this funnel. And we're then going to pour the water and the sand into this system. So what's going to happen? When you pour the water and the sand in the filter, it's like a coffee filter. The solids are going to get stuck. So your sand will get stuck here in the filter, and then all of the water will drip through. And then we will have successfully separated solids from liquids.